Welcome once again to another edition of Fireside Chats. And this afternoon, I am just delighted to be able to spend some time with Lynn Silver. As you're going to hear in our conversation, Lynn is one of the giants of antimicrobial discovery. She has been working in this area, finding drugs and teaching us how to find drugs well, for a while. and and it's really fun to get to share that story and to listen to her insights. So, welcome, Lynn. Thank you for being here. I'm just delighted to have you.
Well, thank you very much, John. I'm happy to be here. I always like to gab about my ideas, and you've always been interested, so that's really a nice thing. So let's
Yeah.
get started.
So,
Yeah.
so to get us started, let's just do a little bit of background on who is Lynn. So, once
Okay.
upon a time, you were a young microbiologist. What happened?
What happened? Well, I was a young microbiologist at Tufts in Boston in the microbiology and molecular biology department as it just after it started. And I had some great professors. Uh, Everyone was interested at that time in cell envelopes, various ways. Ted Park was there, so we had peptidoglycan, and I was working in Andrew Wright's lab, so I was involved in LPS synthesis and also in bacteriophage. So it turns out, even before that, at Brandeis as an undergrad, I worked on phage. I worked on a different salmonella phage at Tufts. I worked on T4, and I've been working on phage for a long time. I was realizing, and it's come back, so that's sort of exciting. I feel like I'm seeing a lot of people just discovering phage, and it's really intriguing. I thought we knew about it a long time ago, and also that the field died a long time ago, but it has come
Yeah.
back. So thereafter, after Tufts, I postdoc in Geneva at the uh, molecular biology department of the University of Geneva. And that was really exciting. There I worked on DNA replication, mostly in E. coli, but also in plasmids, and was sort of involved with people in the early days of plasmid biology. So it was all exciting kind of things going on.
Mhm.
And I went to a meeting in Berlin once where all the sort of big names and plasmids were there, and everyone was arguing about how things worked. And they'd say, well, the way that replication works is blah, 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 blah. And then it turns out everyone's plasmid was different. So no one could agree. Everyone had their own story. Plasmids are all really interesting, but they're not all working the same. And I think that was a discovery that people sort of made, saying, there's a lot of things going on. Anyway, it was a really interesting time. Um, so I, published a few papers back then, actually, much more so than in my thesis, where Tufts is a place, let me go back to that, where people hardly published. You had to have a whole story. You couldn't just publish little things
Mhm.
here and there. So I never published anything from my thesis, which is probably a good
Mhm.
idea. Anyway, but I learned a lot. So in Geneva, published quite a few little papers because we did little papers then. And and then I went to NIH because I wanted when I to come back to the States. It
Ah,
was really uh, hard to find a place that I wanted to be. So I worked in Nancy Nossel's lab working on T4 DNA replication and learning biochemistry. So I basically knew microbiology, genetics that I did a lot of in Geneva, and then biochemistry of DNA replication at NIH. And that was, really interesting. But it turns out when you start making lots of proteins and putting them in big freezers, and there's only one of you, it's very hard to get a job saying, "Oh, I'm going to now start again and make all the proteins I need and redo all of the stuff that I did in my postdoc." So I was saying, what am I going to do? um, and someone from Merck came along and said, "Hey, are you interested in finding inhibitors of DNA replication?" And I thought, why not? I mean, that was a good idea. So that's what I started when I went to Merck in 1982. It was to try. I mean, I had worked with Martin Gellert, DNA, and gyrays, and so I knew a lot of that stuff that was going on. And anyway, what I like to do as a geneticist, I think of myself as a geneticist,
Hmm.
is set up screens or set up, we used to look for mutants by setting up certain screens for that. And so I set about setting up screens for inhibitors of specific targets that I thought were interesting. When I went to Merck, I made a list. I typed out a list based on, probably 1980 J-backed list of all the genes of E. coli. And I said, all of these, here's all the essential genes. I mean, it wasn't that well known, but it was all based on TS mutations and things like that. And I said, there's about 170 genes that look like they should be essential in targets. And then what do we have inhibitors of, et cetera. So when I got to Merck, I said, this is there. And basically, when I learned of what had been going on there at Merck, which was mostly natural products screening, mostly empirical, just, looking for things that kill bacteria. But also, there were a lot of people running. I mean, it was a small group, but everyone ran their own sort of favorite screens and looked for inhibitors of various things. I mean, we weren't only just killing, right? We had some very specific kinds of screens. So for cell wall synthesis, we really looked for all kinds of cell wall inhibitors. And I set up a couple of screens for DNA synthesis inhibitors and other things, protein synthesis, et cetera. But that was sort of my forte, was applying genetics to looking for specific kinds of inhibitors, selective inhibitors of bacteria. And that was fun. And the whole thing is, when I went to Merck, they were very free. They said, do what you want, you'll figure out what we need. So at some point, I was working on a screen for inhibitors of DNA A, my favorite protein at the time, for the initiator of DNA replication. And, so I had a meeting with some of the vice presidents, and they said, well, what will you do when you get an inhibitor? And I said, well, I'll, look for mutants, and then I'll be able to figure out if they're in DNA A, and then and they said, mutants, you want to look for mutants, then you get resistance. And I said, yeah, and that's when it all came out. It started that if you look for specific inhibitors, you'll find resistance like overnight. And so, but I said, no, don't worry. We'll deal with that. But in fact, I still think that, and there's a later instance where this came up, that this is one of the main problems in antibacterial discovery, is that we got very rational in the 80s, basically. And everyone said, well, it's a target. You have to work on targets that and that's because that's what human drugs are looking at. And when I went to Merck, they were looking at HMG-CoA reductase inhibitors. And, it's for human health, they were very enzyme oriented. Yeah. Whereas it turns out for bacteria, there's very few drugs that you use in monotherapy that are enzyme inhibitors. They're mostly ribosome inhibitors or, other kinds of things. Only the beta-lactams and the quinolones, the gyrase inhibitors. Otherwise, they're almost not single enzyme inhibitors. You can use them, but they're not as monotherapy, because that's sort of what I evolved in around 90. I wrote the first paper on that, which was, most of the good antibacterials are multi-targeting. Yeah. And that's important. And then that's sort of something I sang about many times. And I still think it's really critical.
Yeah, there's a paper you wrote that I recall early on reading, that it's your 2006 summary of this concept, but you actually were writing about that well before that, this notion that that single gene targets were tricky. And this paper always stood out to me, that with the multi-targeting by monotherapeutic antibacterials is a good insight. But I had not connected it to the notion that, of course, in human beings, it works just fine to go that way, because we don't mutate the same way. And I can certainly see how the R&D engine would have that idea just would have crept right over into bacteria that had not connected those dots, the 80s and 90s. And that's it's interesting. So Merck had a bunch of natural product stuff too. I mean, again,
Yeah, I mean,
connected
we
to
still
dots.
were working on natural products in those days. So that's all we had. I mean, there were some, there are chemical collections, but we didn't really have good access to those in the screening labs. And most of the screens were actually run in Spain. We had a group of people that we worked with. It was called CBay. Now they're, what are they called? Uh, Modena. I can't remember the name. And so they did our screening. We would devise the screens and test them and run them, and then they would run them all in Spain in sort of not high throughput, sort of medium throughput, with fresh natural products. So that was important. It wasn't a library of natural products. It was things that were grown fresh from isolates, and I think that
Really?
was always important. Yeah. Instead, I mean, we kept libraries thereafter, and they were used for other kinds of screens, for human health screens. But those kinds of chemicals and extracts were always very dilute and, in some sort of solvent. And solvents are not, well, bacteria don't like solvents, they like water. So, the chemist would give you sort of stuff that you couldn't really deal with. But it also turns out that when you do natural product screening, um, you need reasonably concentrated stuff. For example, if you look for penicillins, beta-lactams in fungi or streptomycetes, you'll basically get activity down to about a 1 to 8 dilution, and then it'll go away. So you have to have a reasonably strong concentration, and you can't dilute it too much. Eventually, at Merck, it got down to people using very dilute solutions of natural products, and I would argue against that strenuously, but I didn't have any power, so that didn't help. Um, and that was the other thing that I'd sort of come up with these ideas, and I strenuously, even though I had a reasonable sized group, I had to argue all the time about not that it was silly to just concentrate when I was doing natural product screening. People say, well, the head of research at the time would say, well, you've got to pick a target and you have all the biochemistry set up and you're all ready to do whatever, and pick your favorite targets. And I said, I'm working with natural products. The whole point is you got to find what's there. You've got to take advantage of natural products and not pre-choose it. They're there sort of, I would say for a reason necessarily, but they were evolved that way, and we should figure out how to find novelty. And I still think that's the key with natural products is figuring out novelty. And nowadays, it's sort of devolved into doing all of this. We'll do the genomes of 1,000 bugs, and then we'll look at those genomes and find all the synthetic pathways, and that's really nice. But really, when we looked at any kind of streptomycete or whatever bacteria we're looking at for production, they would produce all sorts of things. Any one isolate, if it was producing anything, would produce 10 things, depending on the medium. And so I have always believed that all of these things were available if you had the right screen. You could find them, and then you would concentrate them,
I mean, so to play it back, you're
yeah.
arguing that there is, if you just have a collection of the natural products themselves, you may end up working with them at a concentration that is too low, or they are dispersed in a solvent-based system that interferes with your ability to do the assay. Whereas an alternative that you have used is you've got, I guess you've got some bacteria or fungi, what
Yeah.
organisms limited to, and you are working directly with supernatants from
Well,
growing orchards.
extract. So they would be done. I mean, you can do, say, methanol extract and then take that up in water or MEK. But the point is that you want to make sure you don't get rid of the water soluble things. And that was important because it turns out that gram negatives really need water solubles. Gram positives are fine with solvent.
Very, very interesting. So, and I don't know whether you've followed it or not. The NCI has a large natural product collection, and I don't know how they prepare their materials, but might be interesting to just take a look at the extent to which their process, would they retain or would they lose water soluble elements?
I don't know that. It's a really, we always just used our own things. I know eventually most groups ended up using libraries like that were in
Yeah,
DMSO or frozen or
yeah,
whatever,
yeah.
and that's a problem. But
Mm.
growing up, fresh extracts that's what the
You can,
Spanish
you
group
can
did.
you can go in a different direction. You can actually control your use of your solvents. Yeah, yeah.
Right, the solvents, but
Fascinating.
also the media. So we grow in all sorts of media for different times. Use a few different solvents. I mean, it wasn't so much that. And then in the lab, I would run little columns and separate, basic from acidic, and do some characterization, because it turns out that in natural products, let's say you're looking for staph. In the beginning, we weren't looking for staph so much. It was always first it was gram negatives, then it became staph. Staph is easy. Like a third of all natural product broths or extracts seem to
Yeah.
kill staph. So not very exciting and not very selective. And when I see papers that I have to review, I'm just very turned off by that concept. That or B subtilis is even easier. So don't do that. But gram negatives are much tougher, and that's where you really need the water solubles because of the way they filter, of the permeabilization. So basically, I got into the idea of permeability working in this area, trying to figure out, how to get things into gram negatives. That was always a sort of interest of mine. And I eventually did write papers on that, a paper. Um, and that turns out to be the other real problem, the big challenge. So when we go through that challenge paper in 2011, it's that if you look at single targets, you're going to get resistance overnight, 95%
Right.
chance. And if you try to find gram-negative agents, you won't, because they don't get in, because the sieving properties of the outer membrane and the cytoplasmic membrane are antagonistic. They're opposite, and
Right.
so you can't get through those barriers. And so that's something that I just kept on thinking all the time and saying. That you have to solve these problems. You can't just keep on going and do in vitro screens for inhibitors. That's just not going to help. And I got into thinking about AI actually, and figuring, well, this should be something where the chemistry should be approachable by looking at neural nets, or I'm not looking
Yeah.
at that, but I figured there's obviously a way to do this. But the key to that, and this is what I'm going to talk about with the AI, is that you can't just lump everything together like all antibiotics and say, what are the chemicals? How are these chemicals alike? And that's what the old literature written by lots of chemists, organic or natural products chemists, but mostly organic chemists, they would just lump everything together, and then they'd come up with, oh, here's something, it's a beta-lactam, or here's a quinolone. But it's very hard to find anything different, because you were lumping everything together. And so my whole way of looking at any of the chemicals, because not only natural products, which are pretty complicated chemicals, but synthetics, which we eventually, started looking at, was you have to bin them correctly. You have to put them together and ask, and I did this in a couple of papers, things that get into gram negatives. So put those together. Things that work in the cytoplasm, things that work, in the periplasm, things that work only on gram positives, not on gram negatives. So those things should give you, and they do. I mean, I have a paper where I just do simple binning based on E. coli, where you can see that there's chemical areas that come up. But it turns out when you look amongst many different bacteria, all the gram negatives, then each species has its own things, because the efflux pumps are different and the porins are different. And so it's very hard to find the commonalities. So this is where when I see papers that are trying to do AI based on a broad set of learning things, I can't figure out how they could come up with anything unless there was a lot of deep data that's built in that I don't see. And I'm not aware of that. So
And
when
so,
this
so,
latest,
so to play it back, but before we talk about
oh yes,
the
I'm sorry, just keep on talking.
no, it's fun to listen to you think out loud. But to test my understanding, and here I was listening to that through the lens of an image I have in my head that talks about the diversity of antibacterials. And there was like the project, the COAD project, where the idea
Mhm.
was to look at very diverse chemistry, because the idea was that unlike molecules for human diseases, things that stay in Lipinski space, the antibacterials tend to go all over the place. And so when you were talking about binning, I think you were talking about the idea of binning based on functional behavior, and then looking inside that bin to see, were there similarities that you could identify the compounds, even though they all came into the bin because of what they did, not because of how they looked. Is that correct?
Yes, a little. Okay. Interesting to pick up Lipinski's rules. Lipinski said, I'm not working with natural products. I'm not looking at antibiotics because they're too different, and that's not the point.
Yeah.
But it turns out then that most things that fit Lipinski's rules, work on gram positives. And so those rules are pretty similar to what works on getting through your intestines. So it's the gram negatives that are really not good. And it's also things that get into the cytoplasm that fit Lipinski's rules. It's not things that are extracytoplasmic. So vancomycin doesn't fit his rules because it's not getting through a membrane. And that's really what I was doing. So that's, instead of functional, it's really location of the target and how how a compound is, what barriers the compound has to go through to get to a target. And those, if you bin them that way, then you should be able to figure out some subset. Now, it turns out, again, that's very often species specific. But at least it's, there's, I think there's some real basis to it. I mean, I would argue that. And if I can just bring up this paper that we're talking about, the Boston paper, that is an AI thing where they're really looking at just Acinetobacter actives. Well, fine. Then you should be able to figure things out. And basically, for gram negatives, my general rule is you're not looking at the targets, you're looking at the permeability characteristics. That's what the compounds are looking at. I used to, work when I would consult. So this is after the Merck. I left Merck and then I consulted for little and big companies and grants and foundations, et cetera, for years, now about 20 years. And I would tell people, well, why don't you, make a bunch of compounds that are water soluble and, have these kinds of characteristics, because I had come up with my own, non-chemist view of what things should be. And one of the chemists from a Japanese company, he's a real smart guy, but he said, well, then you would only use the library once. And he didn't like that. So, but it wasn't worth his while trying to make this, because it's difficult to make water soluble things and polar things. And the chemists don't like that. They like working with solvents, except certain chemists who are up for the challenge. And so that's the other key here, is working together, microbiologists and chemists working together. Something that was always very important to me. And at Merck, we were very good at that. And other places, not so good, especially later on when, even sadly, with small companies, there are some places that had good mix. But otherwise, they would, like send it out and get chemists. They would CROs and things that were doing the chemistry. And it's very hard to sort of get the ideas back and forth if you're not together. I mean, I really think working in, I won't say teams, but just groups that talk to each other.
Yeah.
And that's really an important consideration, is that I think things get broken down, and with high throughput and the way things sort of have flown, uh, things get broken down and put into these high throughput things that really don't ask the right questions. That's why I say go back and ask the basic questions and see if there's a way we can figure this out. And if you're going to do AI, figure out how to ask the question correctly. So lately, there's better work on that. I started that. Paul Hergenrother, I talked to when he started that, um, and so people are coming up with better analyses of how to get things into gram negatives.
Yeah, I thought it was interesting in that second paper from the Harvard group that they had evolved. You know, their first paper basically discovered
Oh yes,
uh,
Hallison.
it covered a poisonous kinase inhibitor. It's fine, but
Correct.
they've learned, and it's just really interesting to see this next one, you know, incremental, stepwise, and this notion uh, of how you develop the breadth of the compounds, sort of the knowledge base that feeds into the. And I'm not nearly a good enough program to know exactly
Yeah, me
what
neither.
it does, but you know, the theory that by looking at structures and linking things together based on functionality, it's just, you know, fascinating to see where they've gotten to with that. Yeah, so this next their fourth paper, it really, it did seem to be an improvement on what they had done previously, and so I've, you know, got my fingers crossed for them. I don't know that it'll ever be Star Trek, but
Well,
maybe not this year, maybe next year.
well, I think, and that's what they led to, and that's actually what
Yeah,
I had noted to you was you can do it in layers. You can add on another layer of modeling, and what they have to do is toxicity. Toxicity is the real killer. I mean, most
yeah,
drug discovery dies in with tox that
yeah,
you can't recognize early enough. Unfortunately, we don't have the right tests. We have a lot of tests, and every time a drug fails, you say, "Oh, I'll add this test
yeah,
in vitro,"
yeah.
but then something else happens, and so what we really need is a better tox ways of testing, and that should be able to be done AI wise. Just doing that kind of thing, and then sort of imposing that on your primary kind of screening, and also other kinds of pharmacokinetic parameters, because that's you have a lot of points to meet, and just an inhibitor of growth is insufficient, because you have to show that it's selective. And let me just go back to the Abosin thing. Awful name. Um, yeah, it's that they said, oh, it's really nice and it hits this really important target. I mean, we do like the LPS transport targets, or this is lipoprotein target. Um, and that's nice, but their resistance frequency was 10 to the minus 7 to 10 to the minus 8. As I said, that's too much for overnight, as long as they're healthy, hardy kinds of mutants. Um, and and there are ways around that, but that's what drug discovery is. You've got to figure out all the points, and just something that kills bugs, even looking selective initially, is not sufficient. So that's it. It's just that there's hard problems to solve, and people should recognize we come part way. It's like everyone working with peptides, which I think there's a lot of good reasons for trying to get good peptide drugs.
Right.
But there's still, every peptide drug has to pass a whole set of problems, and that's what needs to be done. You have to have a way of going through them and making sure you've taken into account all those different parts, which one should be able to do in a sort of virtual way up to a point. So I'm hopeful for AI, but it has to be applied with an understanding of the biology that you're trying to go after.
Yeah. So if you were going to give somebody advice, somebody's interested in starting in this space, you're going to give them a suggestion on, what 5 papers to read first, kind of where do you start to uh, provide people with some guideposts, and and feel free to pick from your papers or from others. I mean, some of your papers are the ones that I know that I go back to personally. But I'm in the, at this point, which ones do you think of when you want to share insights that, in a way that you think is, clear and succinct? Where do you like to go?
Well, I would give him my papers. Also, O'Neill, he's, at Leeds, he's really good. He has a couple of really good papers based with Chopra. He was there, and then O'Neill has taken over that spot. And it's really good to just go through all of the, listing all of those kinds of parameters that you have to be aware of. And I think that's the key. I think people don't understand well how hard doing this is. It's as I've said before, it's not the money, it's the science. And you can get all the money you want, but that won't give you a drug. So
It won't. Yeah. I mean, and I, in my distillation of it, of the list of target parameters, there's a list of proper antimicrobial agents and chemotherapy. The directions to the authors is a paragraph about you need to show at least some of these properties to prove that this isn't just some dumb poison, but it might actually be a chemical of interest. And
right.
Lowell Young and I wrote that paragraph back when I was an editor, because we're forever getting papers
Oh,
about
I know.
somebody's done an extract from some plant, and lo and behold, look, it kills some bacteria. And
Wow.
that's
Well,
nice.
actually, that's one thing. When I first started working, consulting, and I'd work with all sorts of small groups or academics or whatever, and they'd come and want that kind of information. And not that I would give them my papers, but what I do is I say, okay, I'll give you a list of the things you need to make. So I give them that kind of list, the stuff that you needed to know to sort of qualify a compound to be interesting. In what
Yeah.
ways does it meet the criteria? And it doesn't, I mean, toxicity and pharmacokinetics are really important. The target it hits is not as important. In fact, probably it's like the resistance frequency is more important than the target, I think. I mean, if I'd had to give it that way. The chemists hate it. They say, oh no, you need to know the target because then you can make the right chemicals. And that's true, but a lot of times polypharmacology is good.
Yeah. Well, and let's go back to your roots at Merck for a second. This bit about
Yeah.
natural products.
Oh yeah.
I hadn't realized how deep natural products went at Merck. And given you were there for quite some time, working
Yeah.
broadly in discovery, and I know how hard discovery is. If you think back over your career, give us a summary of where you've been involved with development candidates, and just how long it takes for these things to come to fruition, and the fact that persistence is sort of the magic to actually getting anything done.
Well, it's true. So in the natural product sphere, it went on for many years not finding anything that was new and novel. Everything was, the whole problem with natural products is finding the same things over and over and over again.
Yeah.
So you have to figure out, as I said before, to find novelty. And that's really the key, is to figure out how to find novel agents amongst the many repeats.
Right.
So it took a long time before we did that. So this is sort of later historically when we finally figured it, was to use a set of strains where you sort of underproduced single targets, and then you could look for things that knocked out the underproducers but not the wild type. So we had a whole library of those. And then the way that the boss at the time set it up was that each lab got its own set of targets to work on, which I really did not think was the way to do it. But anyway, so we had one, and we're working on Fab I. And And that's when we came across platensimycin, which is an inhibitor of
Hmm.
BABI. And it was completely new, interesting natural product, and interesting structure. And then we found related structures, and they were target specific. And yeah, you got resistance because they were too specific. And that was the problem. They weren't really developable. They didn't have. It was very hard to get the right kind of chemistry, and so it just ran into so many problems. We could prove it was doing what we thought, and there's a lot of good papers in Science and Nature and stuff in this field, and it was exciting, but it just wasn't sort of antibacterial. Some of the compounds in that type really are probably okay for fatty acid synthesis inhibition in mammals, and so have been looked at in diabetes and other things. But anyway, but those were found through these specific kind of screens that were pretty interesting.
Extending off that, the NCI's Natural Product Library, which we discussed earlier.
Yeah.
The hook that they've put out for the public is there are another 3. They said we looked in detail at 75 of 3,000 possible starting points, and we'd like people to start to come talk to us about picking through the rest of these products, and let's see if there's anything out here that's antibacterial. What advice would you give to somebody who's interested in pursuing that based on what you know? How would you begin to go to the next level with the NCI's product library?
Well, here's the thing. If you look at my papers, and not even mine, but all of the fermenters and the people who are doing the microbiology at Merck, the soil stuff, and Dick Baltz, and if you look at it, you have to look at millions of isolates, millions of bugs, to get interesting hits. And when Baltz sort of tracked through how many cultures were looked at for any of the drugs that have come out, I think imipenem was probably several million. But you have to, you're not going to find things in the first 1,000 or 3,000. And in fact, we always used a sort of sub-library of 3,000 to make sure that our screens did not hit anything. Otherwise, they're garbage. Basically, the real hit rate for natural products, for anything interesting, is 0.01 maybe %. And so you have to look at giant numbers of things, and that's just to find something that kills. To find something specific, well, not something that kills. It's more for specific. As I said, in staph, everything kills. but
arguably their ratio, they started off with 330,000 compounds and wound up with this, so 1% loosely. And so maybe of that 3,000, 30 that are interesting. Yeah.
Well, I mean, but it's pretty easy to go through, and triage thousands of compounds. You have to ask these sort of set of questions, and you could do those things pretty routinely. I mean, say for toxicity, that's really, things like that I would do first. And there are people who have set up target arrays so that they try to look for inhibitors of whatever their favorite targets are. But as I said, I'm not in love with that. When you look at really what kind of things inhibit bacteria, it's the macromolecular synthesis systems. You find very few specific enzyme inhibitors, but you'll find pathways that are inhibited, and you can inhibit them in various ways. And there are target-like things. Not every essential gene is a target for bacteria. So I think all of that 20 years of looking at genome target things was pretty much a waste, and why Big Pharma stopped, because that's what everyone did except us.
Well, and here I thought it was interesting that the NCI screen was a whole organism screen. They're screened against a yeast and 2 E. coli's and 1 Staph aureus, and so they were being target agnostic, which I think is a really nice direct go if they're listening to your critiques.
Well, that's
Yeah.
really what we did, the same thing. And we do
Yeah.
it by using that a small spectrum of bugs. And then, that's one way of looking at it. Aside
Yeah.
from looking at cell wall inhibitors, protein synthesis inhibitors. I mean, I think that approach is actually maybe a better one.
Yeah.
Um,
Well, that's a case where you're looking for a particular class of target. I thought it was interesting that they're sort of
yes, but the
whole
point is,
screen. Nice.
yeah, well, I just think that's where the good targets are. I
I
think
see
if
on the outside. Yeah.
that, you start looking for things that just kill, and especially if you're looking at single enzymes, this is just wrong. It just
Yeah.
there's lots
Well,
and lots of them in the literature, but they aren't drugs.
well, if you've been involved in a lot of the pipeline reviews,
Yeah.
and you're sort of well tuned into the direction of things that are coming. If we sort of look forward, um, where are you optimistic or not that we're going to find really interesting novel, uh, really interesting new classes? I mean, sort of what's your crystal ball say about new classes? So as a variant on that is, what's your R&D bucket list of stuff that we ought to be
Right.
doing as a community in order to get us to this, to a place where we actually have some interesting new drugs?
Okay. So I'm going to give you my spiel on that. It's go back and look at all those single target inhibitors and then give them in combinations, just like we do with TB and HIV and HCV.
Hmm.
No one has done that in a reasonable way. They've looked for combinations of all the drugs we have, but they haven't looked at combinations of the single target inhibitors. That's what happened in TB. Every TB drug falls to a single mutation, so you need a lot of different drugs. And same thing for HIV and HCV. We've never approached antibacterial, sort of standard bugs and looked for that kind of inhibition. And I mean, I had a paper. It's in a, what is it? I can't remember which book it's in. It's a journal article where I list, maybe it's 4 years old, but it's, a list of all of the single target inhibitors, and then maybe there's that at the time, and now there's a lot more, probably 40 or 50 different enzymes. So if not all of them are in the same bug, but even say in staph, where most of them can work, but there's also gram negatives. And I still think that's got to be looked at in a reasonable way. I mean, I get people to say, yes, that makes sense, but no one's been doing it. Sometimes George Gisano has pretty much bought into that concept, but few
So
others.
to play it back, you're saying not quite embrace the notion that single targets have a high rate of resistance, but deal with it by saying,
Yeah.
okay, if I can find, if my drug is actually a combination of 2 or 3 single target inhibitors, just go on and a priori commit to a TB like
Right.
approach of looking for several things. And there are there are past evidence
Yeah.
of single target series that haven't been fully utilized. So you
Oh,
could actually
many.
some known materials. Wow. Okay.
Yeah.
We'll find the link to your paper that's got the list of those things and stick it down
Yeah.
below.
Okay. Good. Yeah. I mean, it's at this point old, but that's I've been talking about that for a long time. That I think that's where I would try to spend my time.
Interesting.
Yeah.
Interesting.
Because I really am not sanguine about finding new novel. I mean, there may well be new novel classes, and especially amongst natural products. Synthetics yeah, you might as well look. And doing this sort of layered AI way is not crazy. And synthetics are interesting, and the chemists like to work with them, and they don't like to work with natural products. And so, I think there are ways of approaching that, but I guess I'm of the opinion that the good stuff has been found, and it's going to be hard to find new stuff. But not impossible. I mean, I'm still hopeful, but it's not going to be single target inhibitors by themselves. And so all of that work has been sort of left sitting. And I think it would be good to make use of.
Well, I mean, it's interesting to get that perspective, and you look at like what's coming in the Carbex portfolio. There's some very, very creative stuff in there, but you know, as you and I both know, most of those molecules will turn out to be toxic in one way or another, and
Yeah, I
that's
mean,
just
that's when
for
I
that.
look through that list, it's just frustrating to me. And I look through all of these lists, and enable, the new enable starting. Yeah, I mean, they're interesting things. Some of them are hopeful. Um, and I guess if you think about different ways of screening for stuff, that's fine. And it's always been my experience, and I guess everyone at Merck used to think that you can you can look through the same library over and over again if you have a good, interesting screen that comes up with new novelty and gets rid of the knowns. And so, and there are ways of doing that. That's why when we did screening for these, underproducing versus wild type level, what's the name of the company that started with an E? San Diego. Anyway, that was their thing, and I thought, well, that's great. We'll just do 20 different strains and then find anything that was specific. And Merck really got into looking at that. They had a whole screen based on lots and lots of targets, so they
Hmm.
could do that all at once. And they found things. They found all sorts of after I left, and they found interesting compounds. Which actually there's a small company that's developed working on those now. But always they run into resistance.
Yeah.
Um, so
Anything's
peptides. That's what peptides, or maybe oligopeptides, have a possibility of binding to several sites at once. So a single mutation doesn't give you resistance, and that's the kind of thing I'm thinking of. Is that I really think that's been a major block. I don't think anyone else has. When I was working, with the Pew, we set up this roadmap for antibacterial discovery, and one of the big ideas, I wanted to get in there and did was on permeability and how to look at getting things into gram negatives, but they didn't want to work on the resistance part, which I thought was very important. Um,
right.
and and I felt that was a mistake that we left that off, because for some reason I just think that stopped so many things. I mean, maybe toxicity in the end stops more, but even early on, we don't know enough about in vivo resistance development. I mean, we don't have good models for it.
Yeah.
We don't have good, and that's something that we really need. So,
Interesting.
yeah,
Interesting to think about how that might go. Well, from a big picture perspective,
yeah.
you have you enjoyed what you've done? And would you advise a young microbiologist to do it again?
I don't know. I loved it. I really enjoyed this. I mean, it brought together all these different parts. It's really setting up screens, and that whole genetic approach to things. It's just so satisfying, and I really loved doing that. It's very detective-like kind of thing. And then I learned a lot. I learned chemistry, and well, to some extent, to be like not an idiot when I had to talk to people about it. Um, and so I learned about that. I sucked up things that I learned. I remember when I first went to Merck and would see pictures of carbapenems and beta lactams, and they just put up screens and screens full of those, and I just couldn't understand them. But eventually, I got pretty good at looking at chemical structures and figuring out like what they were doing, or you know what kind of properties they had, and that was very interesting. I mean, I liked it, and I think the whole thing was very intellectually pleasing. I had the chance to really get about, to do a lot of different projects, or to be involved in a lot of different projects. And if I had to just pick one, it would have been really hard. Um, and luckily, at times, I had a reasonable sized group doing all sorts of different things. And that was, fun. Then I could be involved in a lot of things. And you're going to come up, you're going to succeed so infrequently. It's really important to do many things.
It,
Um,
uh-huh.
and so that was that. Otherwise, you just always fail. So
Yeah.
you have to balance that. I mean, this is a very, it's not like um, oncology where they have, there's a million different hits and different new kinase inhibitors, and they're all in the clinic, and we have
Yeah.
nothing.
Well, and the power of a good group of collaborators, I think your career really speaks to that. I mean, over and over,
Yeah,
in this conversation, you have pointed out times when you've worked with one person or another, and that's actually was the collaboration that made the difference.
yeah.
And I guess I think that is you know a young person in this space, there is reason to get into this area.
Oh, it's fun.
Yeah, there we, yeah, it it is fun, and it can be very, nothing's more satisfying than actually making a contribution to a drug, and knowing that no one will ever know your name, but your fingerprints are it's on your fingerprint. It's on one corner of that molecule somewhere, and that's actually really quite satisfying.
Right. No, and yes, I didn't talk about some of the other ones that I worked on, but yeah, that there was always interesting stuff going on, and sort of interim discoveries that at
Yeah,
least you could publish on, which,
yeah.
at least at Merck that was appreciated that you could publish.
Well, and I say, and you've clearly done that. Your series of publications over time are perspective papers on how to think about doing discovery are foundational. Were foundational for many of us, and will continue to be so. So for that, I am quite grateful. Well, we're going to need to draw it to a close, but before we do so, I'll actually just in closing, any other big thoughts that you've got for us about about discovery and directions, topics we haven't covered that we should have covered, in terms of ideas that might spark a career.
Read the literature. Um, I think that people starting out in this, and especially based on, if you look at Twitter, say science Twitter, and people talking about, oh, I found this inhibitor that kills bugs. I mean, just like you said, something that kills staph or something that kills whatever. You got to learn what you're looking for, and what the ropes are. You really don't be naive. I mean, everyone's naive in the beginning, but the point is there's a lot in the literature, and whereas people think, oh, big pharma never wrote anything. Well, that's just garbage. We certainly did. And it just takes, you just have to look it up. Do you think that a lot of academics follow up on things? No. So there's a lot of
Yeah,
papers and stuff that you can find, and there's reviews of a lot of things. So
yeah,
I think it's really up to anyone in getting into the business is to really pay attention to the history. That's where you'll see what worked and what didn't work. And yet, as I said, you might come up with an interesting way of screening that is new enough that you'll find something new, even with the same libraries. And so that's the challenge. The challenge is finding something new and novel and meeting all these criteria, but you've got to learn what that is and what the likelihood is at any stage, the
yeah,
probability of that what you're looking at is interesting.
yeah.
I don't know
But
if
it's a
that's a big.
career worth pursuing, clearly so. Well,
Oh yeah.
um, yeah, yeah, it is fun. Well, Lynn, thank you, and thank you for your comments. This interview was sparked by your note to me about the AI-based discovery, and it was just fun to have an excuse to sit down and chew the fat with you about drug discovery and think again about the perspectives on it. And I say I did not appreciate the depth of the natural product work that you were involved with back at Merck. And now I understand why you wrote all those papers about natural products. I get it now.
Right. Well,
Re-
that was
re-
really what all we did for probably
yeah,
10 of the years I was there.
fascinating, absolutely fascinating. Well, I shall look forward to seeing you at the next antimicrobial jamboree. Not quite sure where that will be these days, but now that it's post-COVID, occasionally we are traveling. And I shall hope to see you somewhere down the road, maybe one of the meetings in Boston this year.
Conceivable, but I'm still immunosuppressed, so I don't go anywhere to those meetings yet until they get a nice set of antibodies that I can take. That's new.
They can protect you.
Yeah.
Well, fingers crossed for that. And again,
Thank
thank you for taking the time to contemplate the universe and points all points across it this afternoon. So you take care of yourself, and I'll see you at the next stop.
Okay. Very good to speak to you, John.
Wow, what a treat to get to spend time talking with Lynn this afternoon. An extraordinary career, depth of experience, synthesized, published for your reading entertainment. I just can't recommend strongly enough that you take a look at her papers and think about the insights that she's sharing for us. And what's that thing I'm supposed to say?
Like, comment, and subscribe for more.
That's it. As always, like, comment, and subscribe for more. See you next time.